Reproduction. A. Asexual reproduction. Binary fission. Asexually, bacteria reproduce by binary fission. In this process, the bacterium, which is a single cell, divides into two identical daughter cells. Binary fission begins when the DNA of the bacterium divides into two, replicates. The bacterial cell then elongates and splits into two daughter cells, each with identical DNA to the parent cell. Each daughter cell is a clone of the parent cell. B. Sexual reproduction. Sexual reproduction in bacteria not involved true mating, so it refers to parasexual reproduction. It takes place by following three processes. Conjugation, transformation, transduction. Conjugation, the type of sexual reproduction in which genetic material is transferred from one bacterium to another bacterium through the conjugation tube, is called conjugation. Discovery. The process of conjugation was discovered by Joshua Laderberg and Edward Tetum in 1946 in E. coli. F. plasmid, fertility factor that to be transfer. Donor bacterium, F+, such bacterium that have an F plasmid. Recipient bacterium, F, such bacterium that do not have an F plasmid. Transformation. The type of sexual reproduction in which genetic material is transferred from one bacterium to another bacterium through the foreign DNA is called transformation. Discovery. The process of transformation was discovered by Frederick Griffith in 1928 in Streptococcus pneumonia. After Griffith, Avery, MacLeod and McCarty gave principle of transforming while work on transformation in 1946. Mechanism. The mechanism of transformation is understood by the factor competence. Competence. The ability of a bacterium to take up foreign DNA is called its competence. Such bacterial cells are called competent cells. The competence may be natural or artificial. Usually, natural competency is rare and found in some bacteria. In artificial competence, cells are made permeable to take up DNA. This can be achieved by two methods. Heat shock method. In this method, cells are treated with Cacl2 and then briefly heat shocked at 42 degrees Celsius for 30 to 120 seconds. It enters the DNA into the cell. This method, cells are briefly shocked with an electric field of 100 to 200 volts to make holes in the cell. The DNA then enters into the cell through these holes. Transduction. The type of sexual reproduction in which genetic material is transferred from one bacterium to another bacterium through the phage virus is called transduction. Discovery. The process of conjugation was discovered by Joshua Laderberg and Norton Zinger in 1952 in Salmonella species. Types. There are following two types of transductioner: generalized transduction, specialized transductioner. Generalized transduction. It occurs in lytic cycle of bacteriophage. Mechanism. DNA of bacteriophage enters into the donor bacterium. It replicates here and develops many copies of DNA. The bacterial DNA is broken and some pieces of it enter into the viral DNA. Then, viral DNA comes out by bursting the bacterial cell. This viral particle then enters into other bacterium, recipient, and transfer the DNA of donor bacterium into it. B. Specialized transduction. It occurs in lysogenic cycle of bacteriophage. Mechanism. DNA of bacteriophage incorporates into the donor bacterium. It replicates along with the some part of donor bacterium and develops many copies of DNA. Newly formed DNA is combination of both bacterial and viral DNA. Then, viral DNA with some part of bacterial DNA comes out from the bacterial cell. This viral particle then enters into other bacterium recipient and transfer the DNA of donor bacterium into it.